Time we get started, are there any announcements for the good of the group? All right. Well, today's speaker is Brett Shelley. He was born and raised in all that in Pennsylvania. And he went to college near D.C., Hood College, where he got his bachelor's and in biology and a master's in biomedical science, which was AKA plant pathology, where he worked in Phytophthora. And since that time, he took a few years off to test out the world and came back to academia because he didn't like it out there in the world. And he's now working on his PhD in the Pilo lab. So today he's going to give us some
So I'm going to talk about recharacterization of amino acid permease 1 in Arabidopsis and what this means as far as a new role in amino acid homeostasis. So to start off, I think it's pretty evident that plants need nitrogen, that if you don't supply them with nitrogen, they don't grow. And we need nitrogen because it's an essential part of the amino acid skeleton, and without amino acids, you can't make the functional units of proteins. So we can take nitrogen up in three ways in the roots, either as nitrate, ammonia, or amino acids, and regardless of which, so nitrogen here is going to be taken up by the roots and translocated to the leaves where the amino acid synthesis is going to occur. And then from there, we can translocate it to organs or into the seeds. So amino acid metabolism is really important for the plant as far as how to allocate the nitrogen available in order to make the proteins that we need to function. And plants have the ability to control this and direct where they're stored. So in order to make amino acids, you need nitrate, but you also need the carbon from the TCA cycle. And once you make the amino acids, where's my pointer? You can either make proteins, and then they can go either into the amino acids can be stored, or they can be used for secondary metabolites. All right, and this all depends on what is available and what the plant needs. And so we can manipulate this, right, and tweak the pathways to really make more nutritious food. And the question then is, how does the plant know whether to allocate amino acids for storage, or to go to proteins or secondary metabolites? And there's this balance between that, and that's what we're trying to study. So I think it goes to say that nitrogen availability affects root architecture, right? This is a classical example showing you that when you add higher concentrations of nitrogen, right, you see increases in root growth. We see this in barley, we see it in corn, we see it in Arabidopsis, right? And this is part of this large complex network in which nitrate is involved in this complex signaling pathway in which different hormones control how the roots develop. And so you can see that the budding of a lateral root is controlled by nitrate from the Zhang paper. Right, but then we're interested in studying amino acids and how they control plant metabolism. And we see that glutamate affects root architecture. So from the cartoon, you can see that when you add glutamate as opposed to just nitrate, you lose root elongation and you see this increase in secondary or lateral roots, right? And that's even evident here in the actual graphs in panel 1 and panel 2, right? Above or below the dotted line, we're adding in different concentrations of glutamate. So where you see the plus, right, we're adding in glutamate. When you don't add glutamate, the roots naturally you can see the primary root and the taproot. But when you add it below in this area where the plant would naturally be growing, right, we no longer see root elongation, and we see that there's this branching and increase where it has no effect above the line, right? So we can even see that here, right? When we look at the root tip and the distance between the root tip and the first root hair, and that in the presence of glutamate, right, the distance from the root tip to the root hair is markedly decreased as opposed to just a nitrate condition, right? And it's been proposed that there are these sensors and receptors that control this mechanism of inhibiting root elongation and promoting the root budding. This is the GLUR receptor. right? So then the question becomes, and this is where I'm trying to go with this, is how do plants sense amino acids? Right? There are sensors that exist both in the cytosol and in the membranes, and potential sensors then include P2, the TOR pathway, right? The glutamate synthase, GNC, and this comes from other organisms, right? Well, we know how amino acids are sensed in other organisms, right? As I mentioned, the GLUR is postulated by some groups, but there really hasn't been advances in the study of this for a while, right? So does that open up that there's other sensors that we might not necessarily know about that aren't in this? And what does that mean? What sensors are there in plants, right? Because these are all from other systems, and some of them are from plants, but there's a wide breadth that we can study. So the mechanisms for sensing, just generally, right, you can have a receptor in which it binds an amino acid, and then there would be a signal transduced from a conformational change in that protein. Or, if it's a transporter, we can transport the amino acids across, which requires a cytosolic sensor that can then give a signal to regulate metabolism. So, right, we can either bind it directly and then give a signal without transporting, or we require the transport and then a signal. So one way we can look to see how amino acids are sensed, right, is we can see amino acids can be toxic to plants. When you apply toxic concentrations of amino acids to plants, you would expect them not to be able to grow, right? And what this means is that, well, they die. And when you have a resistant mutant, right, this means that something is wrong with the metabolism in the plant. And what we see is that plants that cannot sense amino acids should be resistant. So from a toxicity screening for phenylalanine, we found, or we didn't found, but the Falls paper here found that there's this phenylalanine insensitive growth, or PIG1, from a toxic concentration resistance screening of phenylalanine. And basically what happens is that there's major differences between PIG1 and wild type occurring in the shoot metabolism, but we don't see differences in uptake and translocation of phenylalanine. So the hypothesis here is that phenylalanine metabolism then, right, is increased in the pig, which means that there's less phenylalanine in the cells. And if you have less phenylalanine in the cells, right, it's less toxic. And then if it's less toxic, this means that the plant can grow, right? So if you don't have amino acids in the cells and in the plant, well, it should be resistant. So there was really there's no publication concerning which gene is associated with the PIG1 mutation. So a couple years later, Lee's group found that when you, so Wong's group found that in 2017, when you do another amino acid toxicity screening for proline, right, we find this pre1 mutant. So this is a proline-resistant mutant, and what they found when they looked at the genome, it actually localized, there's part of this AAP1, it's to the AAP1 gene. Similarly, when we did, there was Lee's group did a toxicity screening for phenylalanine resistance, and they found that the resistance was in AAP1. And you can see that the wild type and the control and the mutant are both able to grow, but then only really the wild type, or the AAP1 mutants are able to grow on toxic concentrations of amino acids. So we now have a potential gene that is controlling this resistance to toxic concentrations of amino acids. So the characterization of AAP1 was first done in 1993, and it was characterized as an amino acid importer of neutral amino acids. And you can see that it's expressed in the root tip based on the GUS staining. So GUS is just a reporter that shows that it's being expressed in the root tips. And then if we look at the GFP, so the fluorescence, right, from the cross sections, you can see that it's in the root tip, and it's also in the cortex and the epidermis, and then just blown up for you there. So the current model then for understanding how AAP1 works, right, is that we're seeing it in the root tips and in the epidermis, right? An AAP1 knockout means that you wouldn't have transport, which means you don't see amino acids in the plant and in the cells, which means you should have resistance. But I'm going to go through and show you some data from our lab that really challenges this working model and suggests that there might be something else going on that's not holding true to this model. So our lab has done similar amino acid toxicity studies, right? And what we've done is we've screened EMS mutants, and we've come up with several mutants of AAP1 that were isolated, and these are point mutations. So AAP1, here's the cartoon showing you that there are 11 domains, and we have these different mutations on, and you can see that they're spread out from one another, so they're not all localized to the same spot. So how do all of these different mutations affect the function of AAP1? And I'm going to talk about the 4 in black, and then these 2 others will kind of come into play a little later when I get to the end of the talk. So from our screening, right, we did the toxicity, so you have the toxic concentrations of amino acids, right? You see the AAP1 knockout is resistant to all of these amino acids, and then it's able to grow on the control media as well as the wild type here. However, the wild type is not growing, and it's susceptible to death based on the toxic concentrations. But then all of our mutants here seem to be resistant to the toxic concentrations. So then we can look at transport activity of the different proteins, right? Are they functional? So you can use yeast to do this. So here's a yeast that basically doesn't have any transporters in it of amino acids. And then when you have the wild type AAP1, right, we're able to transport amino acids. But then we can look at the different mutants and are they able to functionally transport? And we see that 2 of them are able to transport, and this was obtained from an undergrad in our lab, Fiona. And what you see is that this is the radioactive counts over the time course. And so, as I mentioned, these 2 mutants are transporting, whereas 2 others are not. So you can start to see here that proteins are transporting, but this doesn't mean anything yet. Right, so then we want to start to look at what's going on in the plants. And if you recall from the pig paper, right, they suggested there was a deregulation of metabolism. And what we see is that 1501, 16F1, and 17X1, and even 20AB4, right, compared to the wild type, you can see that the amount of amino acids that are free and not bound to any proteins are markedly higher, right? There's the accumulation of glutamine and arginine in all of these mutants that is different from the wild type. So then, to summarize the last 2 graphs, right, we see that all of our mutants are resistant to toxic concentrations of amino acids, right? So this is what you would expect, that if it's not transporting, then it's resistant. However, from the yeast, right, we see that 2 of them are able to actually transport. So now this is starting to go against what the current model says, right? And then we can see that amino acid content is altered. So this just means, right, score of 1, that's based on what we got from the graph. This is 7 times higher in 15L1 for the amino acid content, 4 times higher, and so on. So my question with all of this then is, why are some mutants able to grow on toxic amino acid environments, but the protein seems to transport normally, right? Because the model says that if you can't transport, you should be resistant. But we're seeing some that transport. So then we wanted to look more of what happens in the plant. So we started to study amino acid uptake by the roots. And so here I'm showing you our setup, right? We have a divided plate in which we put radiolabeled glutamine on the bottom, and then we can observe the amount of radioactivity in the leaves, or in the cotyledons. So the uptake is reduced in the AAP1 knockouts, right? If you can't transport, you shouldn't see any amino acids, the radioactivity being taken up by the plant. And we see that for 2 different concentrations here. So this is just a ratio of the counts per minute over the exposed root length, right? And you see that the knockout, or EH1, is significantly less than the wild type. But we're not concerned about the knockout in the wild type. We're concerned how do our different mutants respond to this. And what we see is that AAP1 mutants are differentially affected to glutamine uptake. And what you see is that, again, we replicate that the knockout right, is significantly less than the wild type, but then we see that 16F1 is actually significantly higher than the wild type. 17X1 transports like the wild type, and 15L1 is unable to transport. And here, this should, right, 15L1 was able to transport in yeast, but 17X1 was unable to transport in yeast. So we're going to redo this to make sure that what we see between the yeast and the plants is consistent and that there's not something different happening in the plants. Right, so the current model, again, right, AAP1 knockout has no transport activity. Therefore, you have less amino acids in the roots, and that should confer resistance. But here I'm showing that some of the mutants are able to transport, and they have resistance to toxic concentrations of amino acids. So then, could AAP1 be involved in amino acid sensing, right? What I'm showing is that amino acid resistance is not dependent on uptake, right? And that we see that based on the amino acid contents that there's an affection, or the metabolism is affected, right? And that the role of AAP1 is not directly compatible with sensing as a transporter. So we need to test if either nitrogen or amino acid responses is affected in mutations of AAP1. So what we can do then is understand which genes are differentially regulated by organic nitrogen or inorganic nitrogen. So right, if you have nitrate, the pathway is the nitrate is transported. You can have a sensor here from the transporter to say this is the level of nitrate in the cells. Right? That's then reduced to nitrate, and then prior to the GS, you can sense how much ammonium is present in the cells. And then the GS converts glutamine or glutamate to glutamine, and then you can have sensors that respond and control protein synthesis. However, we have inhibitors that would block this, right? And then if you inhibit the inorganic nitrogen pathway, right, you would lose any signals that would be derived from glutamine or glutamine. So then if we study AAP1 and the transport of glutamine across, right, we should, if we're getting glutamine across the cells or the membrane, right, we should be able to see the reinduction of, we should be able to see responses to glutamine and other amino acids in an organic nitrogen response, right? So we're trying to find out what are the genes and signals that are responding to glutamine and organic nitrogen as opposed to those from inorganic nitrogen. So Gutierrez in 2008 looked at genes that would be induced by ammonium nitrate, and then those which would be induced by glutamate. And what we see is that when you see red, this is the high expression of those genes. And when you inhibit ammonium nitrate and the GS with the MS6, right, you lose the expression of those particular genes. And then when you add in the glutamate, right, what we're seeing is, in this case, we're trying to find genes that are induced by glutamate, right? So these are all genes that are induced by ammonium nitrate, inhibited with MS6, and then induced with glutamate. So we have, I think it's 6 candidates, 7 candidates that might be potential targets of glutamate sensing and glutamate signaling. Right, so we wanted to look at our wild type. So we repeated this and looked to see, all right, can we replicate this and how do our plants respond? And we see that four of the genes are induced with ammonium nitrate. When you add in the MS6 inhibitor in the wild type, right, we lose the expression of those genes. And then when you add in glutamate plus the inhibitor, right, there's some induction of those genes back to the normal levels, right? However, what you see in the AAP1 knockout is that it is inconsistent with the wild types that would say that something is different in the knockout. But then what about our mutants, right? We see that in all of the different mutants, these are, all the 4 genes are induced with ammonium nitrate, right? They're repressed with the MS6, and then there's some, for the most part, slight induction with glutamine. However, what we see is that if you compare the potassium nitrate response, there seems to be some induction in our mutants compared to the wild type. Right? So, inconsistencies again, what we see is that all of them are resistant to toxic concentrations of amino acids, all of our mutants, right? Some of them are able to transport based on root uptakes. The yeast uptake suggests that some of them are also able to transport. Right? We see altered amino acid contents in the mutants, and we're seeing wild type like gene responses in the mutants. So how do we explain this? And the only way that we can is that a protein AAP1 or a protein also has to have a sensing function. So it has to be a transceptor in that it has to transport, but it also has to be able to act as a sensor, right? And I mentioned PIG, right? PIG is affected in AAP1. So we found this, that the 17X1 mutation is actually the same as the PIG mutation. And AAP1 mutants then can have a deregulated metabolism. So, different sensing options then, right? Well, we know that AAP1 is able to transport. That's how it was first characterized. So that would say that the receptor is not a viable option at this point. However, AAP1 was first characterized as that transporter. So if you have transport, right, you should still see signal. But it's conferring resistance, and there's altered metabolism, which means that there has to be some other role involved in that, because the working model, right, then would say that if you're transporting amino acids, you should confer. If you don't transport amino acids, you should have resistance. But we're seeing amino acids be transported with resistance. So that would mean that the effects have to come from the protein itself and not necessarily some cytosolic component, right? Because we're not actually mutating this. Our mutants aren't involved in, the sensor isn't mutated here. So the goal of my experiment then, right, is to really describe the phenotype of each mutant, right? We want to see how amino acid metabolism is altered in the different mutants, right? We're seeing that with what we have, but on a larger scale, what does this mean for the entire plant? Do we see amino acid contents in the shoots, right? Is the root architecture affected in the same way that we saw with the early studies on this, right? Do the mutants respond the same way as the wild type or a knockout, right? Then we also want to look to see where AAP1 is localized at a subcellular level, right? So as I mentioned, it's expressed in the root tip and in the cortex. So what we're in the process of doing is creating cell type specific promoters that would control where AAP1 is being expressed to see is an AAP that's expressed in the root tip and the mutants, does that have a different effect on amino acid metabolism than say one that would be in the cortex or the epidermis in the elongation zone and the maturation zone, right? And then we can look at responses to amino acids and nitrogen, right? We only have qPCR data for 4 genes, but what other genes are responding to this, right? Once we sort of flesh out how we're studying amino acid metabolism, we can look at RNA-seq data to see the entire library of genes that might be upregulated or downregulated in the mutants, and compare between them if there's an effect on certain genes that might be targets for the sensing mechanism. And then, right, if we understand how AAP1 would be signaling and sensing, right, then we can start to elucidate the pathway. And even if we don't prove this point that one of the mutations, right, so our goal is to say that one of the mutations is able to uncouple sensing from transport. And even if we don't prove that AAP1 is a sensor, right, we'll know more about the pathway in which amino acids are signaling. So one thing that's been implicated in amino acid signaling is the mTOR pathway. We've seen this in mammals that mTOR interacts with a RAS protein, which is a GTPase, that's able to transduce a signal. And what you see is that nutrients and amino acids in the mammalian TOR, or mammalian system, all feed into TOR. Right? We don't know as much about the plant TOR system, and you can see that nutrients and amino acids are feeding into the system. And what we find, based from Dong and Montaigne, is that inhibitors of the pTOR system have effects on root elongation, the activity of the meristem, and in root hair development. Right? So we can see that there are changes in root architecture based on activities to the TOR pathway. So the conclusion then from this is that the AAP1 mutants that we have exhibit this deregulated metabolism, right? We see that with the amino acid content, right? They're resisting even though they're transporting. So the only logical explanation for this is that AAP1 has additional roles in sensing that are not exclusive of a transporter only, right? We need to study how amino acids are used by the plant following this. So as I mentioned, right, the TOR pathway might be involved. We have, as I showed in the diagram at the beginning, right, there are two loops. The lab has actually identified via yeast-2 hybrid that there is a RAS homolog, REB, that actually interacts with AAP1. And this might be the intermediary as part of the sensing mechanism between AAP1 and the mTOR pathway. Right? And our ultimate goal then, right, is to understand amino acid metabolism so we can create plants with this engineered metabolism to control the allocation and the protein content, right, to increase that, to make more nutritious food. And also, right, we can then control how much nitrogen we're adding and lower fertilizer application. So with that, I need to thank the Pelot Lab, Kasia and Unadi, who have taught me the techniques, and then Rajan and Shelton, who did a lot of the preliminary work on this, and then Fiona, whose data you saw for the yeast uptake, other members, the school for providing the funding and the opportunity, and then my committee members, Dr. Winkle, Dr. Gillesco, and Dr. Beers. So with that, I'll take questions. And is AAP1 a transceptor? That's to be elucidated. John.
So can you go back to the slide where you showed the increased fold in amino acid content in leaves? There's a table. Is it the next one?
This one?
Yeah, this one. So mutant 17X1 is kind of interesting because it shows high amino acid uptake in the leaves, but it's not showing transport in the yeast uptake assay. What do you suppose is going on there?
Well, the first thing we need to do is make sure that, right, what we see with the yeast data is consistent in the plant, because the 1501 and the 17X1 seem to be switched. So I'm going to redo that experiment to make sure that, first and foremost, what we actually see is true, and it's not just, well, somehow samples somewhere got mixed up. The only logical thing that makes sense, right, is that there has to be some regulatory network associated with the protein. And what we're seeing is that it's not transporting, but there's also something involved in a sensing mechanism.
Yeah, I mean, maybe the sensor is turning on some other transporter as well, but
Yeah.
it's helping to pull it in, which would be consistent with some sort of constitutive sensing mechanism. But that one kind of jumped out.
Yeah, the inconsistency with 1501 and 17X1, I want to make sure that that's consistent with what we see, but the 17X1 is the PIG mutation. So, I mean, they suggest that there is a coupled mechanism, but that should be it should be transporting, right? So if it's not transporting, then that would be the reason, but it's clearly, I mean, there's more, the Ball's paper says that there's more catabolism of the higher aromatic amino acids, which leads to the increase than
Huh.
that. But apart from redoing this to make absolutely sure, I think that's kind of what's going on.
Still early days.
Yeah.
Fair enough.
Is there anybody toxic for producing plants?
Overexpressors. We could have that. I don't show it, but your GDU has all the metabolism too, but that's just that. But I mean, we could have more I mean, we could actually add that in. I don't think anyone's looked at overexpression of AAP1. I mean, I suspect that it would just be toxic to the plant if you imported more amino acids than, right? Because it's probably selective, it probably has some mechanism, but if you just open up the pores, it's probably going to transport. At that point, it might be toxic, but
Well, if
Yeah.
we growing plants in a bad sun acid condition, they become growing normal. We don't really need to add nutrient for growing plants. They can take the nutrition.
I mean, you would expect to see then maybe changes in nitrogen metabolism too. That's kind of what we're getting at, is we don't necessarily know. I mean, I can actually add that in to see how that would affect the roots. Does it cause a change in the root architecture that we don't necessarily know? I mean, I don't think anyone's looked at it, but so I can't postulate what it would do. I mean, I suspect that there'd be some alteration to metabolism that we would probably see either more or less amino acids in either the roots or the shoots, depending on how the mechanism works with the plant to regulate metabolism.
So you're talking in the field?
Only if we just, if we test AAP for producing plants, then kind of like to know whether there's more amino acid taken or because of the homeostasis, we know what producing maybe take.
Well, I know it works for another amino acid transporter, HT1. But actually, there have been some plants constructed that overexpress AAP1. I just discovered it this morning by getting a PhD thesis. It was published in 2004. I haven't had time to analyze all the data. But the plants do not look good. That's what I can say. I don't know about it.
All right, any more questions? Okay, hearing none, let's thank our speaker.